Hello dear viewer, welcome to my YouTube channel. In this video lecture we will discuss the Watson and Crick model of DNA, structure of DNA and also different forms of DNA. Let's start. Watson and Crick model of DNA. In 1953, two scientists Watson and Crick present a model of DNA known as Watson and Crick model of DNA. The main points of that model are that DNA is double helical structure twisted around each other. Have a look at the diagram. This is the double helical structure of DNA which are twisted around each other, in which the base pairing rules are always maintained and the two strands are complementary to each other. That is, adenine bend to thymine and cytosine bend to guanine, and hydrogen bond are present in between the two strands. That is, adenine and thymine have double hydrogen bond while cytosine and guanine have triple hydrogen bond in between. The both strands of DNA are antiparallel to each other, in which one strand is 3' and other is 5'. While in DNA replication, each strand acts as a template for the synthesis of another strand of DNA. Now have a look at the structure of DNA. Within a single turn, 10 base pairs are present. Here in the diagram, the black arrow shows the base pairs, which are adenines and cytosine, thymine and guanine nucleotides. And one single turn are about 34 angstrom in length. The distance between two nucleotides is 3.4 angstrom. And the diameter of the helix is about 2 nanometer or 20 angstrom. The DNA have major groove and minor groove. The major groove of DNA is about 1.2 nanometer and minor groove are about 0.6 nanometer. Different forms of DNA. There are alternative forms of DNA are present because of different environment and condition. The structure and form of DNA can be identified by X-ray diffraction method. Different forms of DNA are available. They may be natural or synthetic. The natural form of DNA include A form of DNA, B form of DNA and sometimes Z form. These are natural form of DNA. While artificial form of DNA include cDNA, ddNA, eDNA and pDNA, which are not mentioned here. Let's discuss each form in detail. A form of DNA. A form of DNA are natural DNA and are present under high salt concentration and dehydrated condition. This A form of DNA is short and fat as compared to B form DNA. In this form, 9 nucleotide bases are present per turn and the diameter of A form DNA is 23 angstrom, which is equal to 2.3 nanometer. It is right-handed double helix DNA. Their base management is changed, so their major groove is wide and minor groove is narrow and are slightly modified. Next is B form of DNA. B form of DNA is natural DNA and this DNA is present in Watson and Crick model. This type of DNA is found under low salt concentration and in aqueous environment. The diameter of this form of DNA is 2 nanometer or 20 angstrom. The B form of DNA is also right-handed twisted DNA. Each turn contain 10 base pairs. These are the base pairs of DNA, and their length is 36 angstrom or 3.6 nanometer, and the pitch or the distance between two nucleotides is 3.4 angstrom or 0.34 nanometer. Now have a look at C form of DNA. C form of DNA are synthetic and right-handed twisted DNA. This form of DNA are present on higher salt concentration. Also they have 9.3 base pairs per turn. Like aDNA, the cDNA does not have base pairs lying flats. Their base pairs are flats, just like A form of DNA, because it has tilted base pairs. Next is Z form of DNA. Z form of DNA is synthetic form of DNA, and they have guanine and cytosine base pairs only. It is the only DNA which are left-handed double helix DNA, wends to left in zigzag conformation. That's why this DNA is named Z form, and the diameter is about 18 angstrom. Their major groove is almost absent or eliminated. They have only minor groove. It is long and thin DNA as compared to B form of DNA. D form of DNA are artificial or synthetic DNA. This form of DNA is right-handed double helix twisted around each other, and the D form lack of guanine nucleotide. They contain 8 base pairs per turn. E form of DNA are synthetic DNA, just like C and D form of DNA. Also they are right-handed DNA like other forms except Z form, because they are left-handed DNA. They are lack of guanine nucleotide and having 7 bases per turn. Last one is P form of DNA. When this DNA is stretched artificially, P form of DNA comes into existence. This form of DNA is longer and narrow. The phosphate groups present inside in pDNA, and the nitrogen bases present near to the surface of helix, and also they have 2.62 bases per turn. Thank you dear viewer for watching. Subscribe my YouTube channel for upcoming microbiology lectures.